Bismillahirrahmanirrahim. Assalamu alaikum. My name is Areej Fatima from BS Chemistry, Batch 1. Today my topic of presentation is Jacques Dubochet. First we talk about contents. Introduction, scientific career, research, diagram of cryo-electron microscope, and references. Firstly, introduction. Jacques Dubochet is a retired Swiss biophysicist. He is a former researcher at the European Molecular Biology Laboratory in Heidelberg, Germany, and an honorary professor of biophysics at the University of Lausanne in Switzerland. In 2017, he received the Nobel Prize in Chemistry together with Joachim Frank and Richard Henderson for developing cryoelectron microscopy for the high-resolution structure determination of biomolecules in solution. Born. Jacques Dubochet was born on 8 June 1942 in Egle, Switzerland. Citizenship, Switzerland. Education. He has done his BS in École Polytechnique Fédérale de Lausanne and MS in University of Geneva and PhD in University of Geneva and University of Basel. He is known for cryoelectron microscopy, and he has awarded for Nobel Prize in Chemistry in 2017. Scientific career fields. Two fields included structural biology and cryoelectron microscopy. Institutions: European Molecular Biology Laboratory 1978 to 1987 and University of Lausanne since 1987. Thesis. He has done his thesis with contribution to the use of dark field electron microscopy in biology in 1974. Doctoral advisor is Edward Kallenberger. Research. Jacques Dubochet was awarded the Nobel Prize for Chemistry jointly with his American colleague Joachim Frank and his British colleague Richard Henderson for their work on electron cryo microscopy. Their achievement in layman's terms was to develop a technology that allows scientists to examine biomolecular samples under a microscope without having them dry out or lose their natural structure. Their technique consists in vitrifying the water in the samples, for example, freezing them so quickly that they do not have the time to crystallize. The first high-resolution structure determined using cryo-electron microscopy was presented in 1990. A decade passed before high-resolution structures of helical and icosahedral structures were imaged, determined based on analysis of data recorded on film. After the introduction of new direct electron detectors in 2012 to 2013 and the first reports of de novo atomic structural models of smaller single particles such as that of the membrane protein ion channel, cryoelectron microscopy has very rapidly become a major new tool in structural biology. About 6 decades after John Kendrios and Max Prout's pioneering crystallographic work on myoglobin and hemoglobin, and 4 decades after the first developments that laid the groundwork for single particle cryoelectron microscopy, a high-resolution structure of hemoglobin in solution determined using cryoelectron microscopy was presented. Cryoelectron tomography is used to determine the structure of even larger objects, including organelles and cells within the potential. The resolution progression of cryoelectron microscopy illustrated by the representation of glutamate dehydrogenase with an increasing level of detail from left to right. For a protein of this size, 334 kilo Da diameter, the 1.8 diameter resolution to the high right could only be achieved from 2012 and 2013. After an image by V. Falconeri, cryoelectron microscopy extends the possible size range for structure determination in solution from cells and organelles to molecular complexes, molecules, and the atoms that build these molecules. But cryo-electron microscopy is not only about static structures, because sample preparation for cryo-electron microscopy involves instant cooling of solution. The contents of the solution can be systematically varied. Integral membrane proteins may be studied in a near native environment, and the particles may be trapped in structural substrates. For example, while an enzyme catalyzes a chemical reaction. These recent developments will certainly be followed in time by others, both in regard to technology and applications. Perhaps in the future, we will be able to obtain high resolution structural information of molecules as well as to observe interactions and dynamic processes as they happen inside cells or organelles. This progress would not have been achieved without the contribution of laureates. Jacques Dubochet developed methods for preparation of samples for cryoelectron microscopy, studies of biomolecules in water. Joachim Frank developed methods for structural determination of biomolecules from analysis of assemblance of particles in solution. Richard Henderson demonstrated that it is possible to obtain atomic resolution structures of biomolecules using cryoelectron microscopy. And this is the diagram of cryoelectron microscope. These are the websites from which we take the data. This is all from my side. Thank you.